All right. Now, this looks overwhelming. Please don't let it. Like we just said, we have three current domains, right? Been around since early 2000s. Three domains. I wish I could switch these two around because we actually believe the very first living thing that developed on the planet came from the domain archaea. Archaea, archaic, very old, but also extremophile. Ooh, fancy word that simply means lives in extreme environments, extreme temperature, extreme pH, extreme salinity. These are factors that are going to be important when we talk about macromolecules again. Don't worry about it right now. But beginning of life, beginning of the alphabet, A and A, look at that, right? Bacteria, this is your typical bacteria, like your strep throat, streptococcus, your E. coli, Escherichia coli, your staphylococcus. These are the ones that we take antibiotics for if we have a bacterial infection. Archaea, antibiotics will not work for these. Now, antibiotic resistant bacteria still falls under this category. We'll get back to those later, but it still falls under this one. Now, like we mentioned, we've got prokaryotes and eukaryotes. If you're in the domain eukarya, you're going to be a eukaryote, right? You're going to be animals, your plants, your fungi, your protist. The technical definition of a protist right now is you are a eukaryote that is not a plant, not an animal, not a fungi. That is the scientific definition of a protist currently, because scientists right now are really trying to understand where protists stem from. When this all happens, this is when that switch is really going to start happening truly to the phylogenetic tree. It's already in the process, but these types of charts will no longer be a factor. It'll be more the phylogeny, which we'll go into when we talk about evolution. But this is a nice simplified way to understand these similarities and differences between the different domains and kingdoms. All right, cell structure, we're going to skip this for a second because it's a little intimidating and we want to just get nice and comfortable with all these guys. Now, bacteria, archaea, whether it's your true bacteria or your archaic bacteria, right? Don't think that they're no longer around. Like I said, they're still around. These guys are both unicellular. They're made of one cell. Plants and animals, they're multicellular. Well, what about fungi and protists? Well, they have everything stemming from unicellular to multicellular. Okay? Mode of nutrition. We're looking at animals. How do they eat? Troph refers to food. Hetero means different. So heterotroph, you're getting food for something else, from something different. Autotroph, you are self-feeding. Animals have to eat something else. We know this. Plants make their own food. We know this. What about fungi? Okay, let me ask you this question. Do you see mushrooms growing in the sun, or do you see them growing in the darkness? In the darkness, right? Well, if they grow in the darkness, then they can't be autotrophs. That's a nice, easy way to remember it. So these guys have to be heterotrophs. The other three kingdoms, they can go either way. Now that we've gone over these categories, let's go back to cell structure. Animals, we know this from high school bio, they have no cell walls, right? No chloroplast, but no cell walls. Plants, they have cell walls. But what's the composition of their cell wall? It's actually cellulose. Think of celery, okay? So, cell walls made of cellulose, celery, we can eat it, we can't get any energy out of it because we just can't break down that cellulose, okay? That's why there's 0 calories. Fungi, just like that's a cool name, well, their composition of their cell walls is pretty cool too. It's called chitin. You'd think it'd be chitin, but it's chitin. Now, protists, how can you remember these guys? Well, I've got a little story for you. When I was in college, my marine botany course was plants. Now, everything that fell under plants is under protists. The why, I'll explain another time. What matters is it'll help you remember that if they used to be in plants, well, if the protist happens to have a cell wall, it's going to be made of cellulose. Okay? But not all of them have a cellulose. It just depends on which species. Now, cell walls, peptidoglycan. Your bacteria is going to have that peptidoglycan. Your archaea is not. What in the world is a peptidoglycan? It's just a protein with a carb that stems out past the actual cell membrane. And because of these proteins, they come together and interlock, and it's almost like the two bacterial cells are holding hands. And they form these colonies. So even though they're unicellular, they form colonies. And when they're in colonies, when they're in high numbers, our immune system has a harder time fighting off. That's why we have to take the antibiotics for strep, for E. coli, for staph. So how do antibiotics work? Well, not all, but some of them actually work by covering up that peptidoglycan so they can't bond to each other. And now your immune system, instead of attacking a colony of thousands, it might be attacking a colony of 5. 5 is a lot more doable to attack than thousands. So, okay, with our prokaryotes versus eukaryotes, uni means one. We're referring to one unicellular. Multi is going to be many. Your auto, that's your self. Autotroph, right? Self-feeding. So think of food. And hetero is different. Okay? When we think of pro, think of no. Prokaryotes have no nucleus. Eukaryotes, that means true. EU means true, like we mentioned with bacteria. Eukaryotes, they're green because they've got the nucleus. The other thing they have is what we call MBOs, membrane-bound organelles, an organelle with a membrane around it. What are we talking about here? We're talking about mitochondria. We're talking about chloroplast. We're talking about vacuole. We're talking about endoplasmic reticulum, rough and smooth, Golgi apparatus, lysosomes. Your eukaryotic cells are going to have them. But guess what? Your prokaryotes, they're not going to have the MBOs. Okay? Now, your prokaryotes, again, these are your archaea and your bacteria. Right? Your eukaryotes, these guys, that's supposed to be in red, sorry. Right here, the MBOs. Crossed out, that's supposed to be in red. But your eukaryotes, that's your plants, your animals, your fungi, your protists, right? Now, great, we've talked about how they're different. Now we gotta discuss what they have in common. I like to use blue for both, right? So in your mind, you can close your head and go, okay, what was it? What was it? What color was it in? It was in blue, so it's got to be both. Now, when I think of a cell, I think of a room. So the boundary of the room is the walls, the ceiling, the floor. So in a cell, that would be your cell membrane. Okay? Now, we've got people inside the room who do work. That's your workers. Well, your ribosomes make your workers. Okay? We've said from the 8 characteristics of living things that all living things have to have a DNA. So we have to have a DNA. Now, notice we did not say nucleus, no nucleus, DNA. And then last, we got to fill the room up with either some kind of air, or if it's an aquatic environment, water, right? Well, then that would be your cytoplasm. So these are the 4 things all cells have to have. Not mitochondria, not nucleus, the 2 most common mistakes. Cell membrane, ribosomes, DNA, and cytoplasm. All right, 3 domains. We've got bacteria and archaea. They're composed of prokaryotes. Most are unicellular and microscopic. Archaea live in the Earth's extreme climates, such as your salty lakes, your boiling hot springs, right? Just like we said, we believe the first living thing was archaic, being able to withstand that extreme environment when the Earth was starting to form. Eukarya, composed of eukaryotes. We've got our red blood cells right here for our animal cells. Our stomata, what opens and closes to allow oxygen and carbon dioxide in and out of a plant cell. Our bread mold, right? That's one of our fungi, our yeast. And then right here, our diatoms, which is one of the protists. And then here we've got examples such as E. coli, Anabaena, and some of our archaebacteria, some of our extreme organisms. discovery science. In order to describe natural processes and structures, we have to go through science, through the scientific method. The observation, right? This is where we're going to use all of our senses to gather information, directly or indirectly, with the use of tools. Our data, these are our recorded observations using all of our senses. Now, are these numerical measurements? Are they numbers? That's quantitative data. Or is it a description of something? Qualitative data, right? Our hypothesis, this is a tentative answer to a well-framed question. It's basically an explanation on trial. It's a possible answer to a question, right? Now, we're using that, we come up with that hypothesis using our past experience, our available data of discovery science, and putting it all together and trying to come up with what we think is the answer to this question. Deduction, this is a type of logic that we build into our hypothesis-based science when we use deductive reasoning, where logic flows from a general direction to a specific one, which is the opposite of inductive reasoning. Now, deductive reasoning is incredibly important when creating our if-then hypothesis. When we write a hypothesis, we've got to use a deductive reasoning. It's going to have an extrapolation from general premises to specific ones. If this is true, general statement, then so must this be. It's specific. So if all organisms are made of cells, premise 1, and humans are organisms, premise 2, then humans are composed of cells. That is your deductive prediction about a very specific case. And remember, this is what we come up with at the beginning of an experiment, right? Of one experiment. So 2 important qualities of any hypothesis: it's got to be testable and it must be falsified. Basically, once you do your experiment, we want other people to try and prove us wrong. And if they're not able to, then we actually gain credibility because they were not able to prove us wrong. It's really kind of a weird approach, but when we think about it, this is really how we know, hey, we didn't make a mistake, or we didn't use an incorrect assumption. Now, the myth of the scientific method is any chart like this about the steps of the scientific method. The very first time the steps of the scientific method were written down in a chart was actually for a textbook. However, there are certain steps that are going to occur in a certain order, if you will. Or flow would probably be a better word for it. You would not come up with a hypothesis after you've drawn your conclusions, right? If the hypothesis is an attempted answer that we're testing, well, then that's going to happen before we actually get our conclusions drawn. But you could do a question, then research, then hypothesis, or you could do research, then ask the question, then do the hypothesis. It just depends. And sometimes, even after you've done your entire experiment, you realize that part, if not all, of your hypothesis was completely wrong because you didn't go at it the right way, or you had an incorrect assumption. So, or there was a variable you didn't take into account. So then we have to redo things and start over, and that's okay. Science is about getting to the answer, and you're gonna make mistakes on your way to getting to that knowledge, and that's okay. That's why in any class, especially this one, please feel encouraged to try to answer questions when I ask them, because we're on that path to gaining knowledge. So it's completely okay to make mistakes. All right, controlled experiment. This is where you've got an experimental group, which is compared with a control group. We're controlling unwanted variables. We're not going to eliminate them. You can't, especially often in the field. Inside a lab setting, it might be easier to take a lot of them out of the situation, but you don't want to take them all. We need to know what's going on, and if a particular data shows a trend, we need to know what caused that trend. So our dry mix, dependent variable responds to the y-axis. So let's review. This is our y-axis. This is our x-axis, right? So I don't know where this was when I was in school, but oh my goodness, I love it. Dependent variable is what responds, and it's on the y-axis. So inches, height, that's your dependent variable. What's on the x-axis? That's your manipulated, okay, variable, your independent variable. That's what's on the x-axis. That's the months, the site of the actual forest, right? What kind of soil they were looking at. So the height the tree grows, that's on the y-axis. Dry, that's your dependent variable. Types of soil used, That's on the x-axis. That's your independent variable. Rainfall, the amount of rainfall every single month. Those are your, right here, inches of rainfall. That's your dependent variable again. And on the x-axis, the months, that's what we're looking for, the different months. That's your independent variable. A theory is what we come up with after we've done many, many different experiments, right? Again, hypothesis is at the beginning of one specific experiment, but a theory is what we have come up with after many different experiments have done, and it's very broad. It's not specific, and it's after many different experiments, and because we have all these different experiments, conclusions and data, we can actually have evidence to support our theory. Our theory is really our attempt to explain nature, whereas a law is a description of nature. You have to understand, and I know this is still a common misconception, hypothesis does not become a theory, and a theory does not become a law. All right, so does a theory become a law? Absolutely not. You cannot have an explanation become a description. Okay? The why can't become the what. So, hypothesis, beginning of one experiment, very specific to that experiment. Theory, after many different experiments, very broad. Law is a description of nature. And that's the end for this chapter.